Alright, thank you so much guys for coming. So today we're going to do lecture 11. We are already on material for midterm 3. And we'll talk about the nervous system. We'll pretty much just talk about the way the nervous system is organized, and we'll talk about the two main branches, central nervous system and the peripheral nervous system. Alright, and then we'll talk about the two main cell types, the neurons and the glial cells. The glial cells are ready for support, neuroglial cells. And then we'll talk about the types of neurons there are, of which there's 3. You got unipolar with one process, bipolar with 2, and then multipolar, which is the bulk of the neurons, with many processes. We'll talk about that, dendrites and one axon. And then we'll talk about the different types of glial cells in the periphery, which we have Schwann cells and satellite cells, and then we'll talk about the glial cells in the central nervous system. So there's 4 of them: the astrocytes, oligodendrocytes, and microglia, and parenchymal cells. So we'll talk about that and their functions, and we'll also talk about the special characteristics of neurons. Because neurons, once they are made, they cannot regenerate. So they have special organelles that are, concerned with repairing, mostly protein-making machinery in the cell. Which organelle does protein synthesis in the cell? Like we've done before. Which organelle does protein synthesis? Which one? What's it called? Starts with ER. Yeah, the rough endoplasmic reticulum does that. Okay, so in the neurons, they got a lot of free ribosomes and a lot of rough endoplasmic reticulum, and they're called basal bodies because there's just a lot of it, since these cells do not divide. So we'll start first to talk about the functions of each system that will come. So we all know that communication, controlling of all the events in the body, especially to maintain homeostasis. Okay, remember when we were talking about the homeostatic mechanism, we talked about the control center, in that it compares the change in the variable with the set point, and then gives instructions to the effectors like glands and muscles to carry out the response. So in that, we talked about how you need receptors and sensory, right? Neurons to take the information to the CNS, right? And then once it's there, then this is the controlling, comparing to the set point part, where you make sense of that stimuli and then you generate a response. Okay, so we all know about that. So if I ever ask you about functions of the nervous system, you shouldn't have any problems, because this is repeating the same stuff we did for homeostasis and the homeostatic mechanism, right? So integration, of course, is when the sensory information is processed and then a response is generated. And then when that happens, it sends an impulse through motor neurons to the muscles, skeletal muscles, if you need to do some running or something. And I'll show you a good example later on of the functioning of the nervous system, or to the glands to release a hormone, things like that. Okay, and all this is meant to maintain a stable internal environment, right? Which is homeostasis. That's what it is by definition, maintaining a stable environment. Okay, and all the hormones and all that are involved with that are secreted, controlling metabolism and maintaining the skeletal muscles, the heat for temperature and so forth, all to maintain homeostasis. And then, of course, we know that the brain is the seat of intelligence. That's where all the mental activities go. Okay, so we all know about functions. Pretty much, the nervous system is divided into 2 main regions, right? The central part. Okay, so we have the nervous system, right? It's split into the central nervous system, which includes the what? The brain and spinal cord, all right? And then, not South Carolina, spinal cord, okay? And then, what else? The peripheral nervous system. So the peripheral nervous system is the perimeter, all made up of cranial nerves, spinal nerves, right? All these spinal nerves, they are outside of the center, right? And then this peripheral nervous system is split into sensory nervous system, right? Which carries the senses to the central nervous system, all right? And then the motor nervous system, all right? The motor nervous system carries information to the periphery, right? And it instructs the glands, the muscles to do work. And then the motor is split into the somatic nervous system. Remember when we talked about the somatic nervous system, when we were talking about muscles, we said muscles in utero arise from structures called somites. Hence, somatic nervous system controls skeletal muscles. Okay? And then anything else that's automatically controlled is the which system? Anything that's automatically controlled is the autonomic, yes. Autonomic nervous system. Alright? The autonomic nervous system is split into three. Okay? The first one is the parasympathetic. Does anybody want to guess what this one does most of the time? The parasympathetic does what? It slows down, it inhibits. Yes. It slows down, it inhibits. And then the second one is the sympathetic. Usually if you're sympathetic to somebody, you want to add something to them, you want to stimulate them. So the sympathetic normally does what? Yeah, flight situations is the sympathetic. And that's epinephrine, norepinephrine, right? So that's that. And so this is usually resting activity, okay? And because the digestive system is such a complicated system that needs elaborate nervous control, there's an actual branch of the nervous system called the enteric nervous system, which only deals with digestion. Okay? So this is pretty much how the nervous system is organized, and I'll show you this chart again because I need you to know about these charts, and then I'll talk a little bit more about what each of those branches of the nervous system do. Okay? But the main anatomical branches are the CNS and the peripheral nervous system. Okay? So in the exam, normally I ask you to tell me about organization of the nervous system, and I'll ask you to give me that diagram, that organization chart. And then I'll also ask you to tell me what each of those branches does. What is the function of each of those? Okay? I normally do that, so you gotta know that. That's how the nervous system is organized. So we talk about each part now. Okay, like we already said, central is the brain and the spinal cord. And we all know about the skeletal system and the functions of the skeletal system, right? From the exam, what is one of the functions? It's protecting. So these things are encased, the cranium encases the brain, right? So what encases the spinal cord? Yeah, the vertebra. See, they got a hole in the middle. So the vertebral column, the vertebra does that. So they are encased in bone for protection. And then, of course, the peripheral nervous system is the other major division of the nervous system. And this includes, the receptors at the end there, and then the spinal nerve, of which there's 31 pairs on either side of the spinal column, and then 12 cranial nerves, right, that arise from the base of the brain, pretty much mostly from the brainstem, okay, and so cranial nerves, that's all periphery, all right. So those are the two main anatomical components of the major divisions. The peripheral nervous system will detect stimuli, and the receptors will catch it and then send it through sensory nerves, sensory neurons, to the central nervous system, where that information is processed and integration takes place, and then pretty much making sense of the stimuli of that sensory information. That's what the brain does, the central nervous system does. And then once sense is made out of that stimuli or that sensory information, a response is generated. Is that all the time, though, that a response is generated? No. Sometimes the response is not. Because, again, remember with homeostasis, it is maintained within a certain range. It doesn't flatline. So it doesn't always respond all the time. So sometimes the response is not necessary and nothing happens. Okay? So, know the function of each of those major systems and, how this nervous system is divided up. So we move on, right, to the peripheral nervous system. So I want you to know the divisions, and I want you to know which of those parts or branches is divided up. Okay, not only just be able to list them like that, but be able to tell me, no, it is the peripheral nervous system that is divided into this and that. You know what I'm saying? It is the autonomic system that is divided into this and that, and not the somatic, and things like that. You know what I'm saying? So we get the sensory that goes up to the nervous system, to the central nervous system, the brain mainly, right? Okay, passing through the spinal cord. Okay, and this is just that division. But the motor division, the one that gives instructions to the rest of the body, that's the one that's divided up. Okay, so the motor division carries action potentials through motor neurons to effectors like muscles and glands, and so on. Right? So the motor is divided into the one that carries information to the muscles, called the somatic, okay, the skeletal muscles, and then the one that carries to all the rest of the other muscles, mostly smooth, right? Because smooth muscle is in blood vessels, smooth muscle is in the digestive tract, smooth muscle is in the respiratory tract. All those things that are automatically controlled by the autonomic nervous system. And then it is further subdivided into 3 subdivisions, right? One that is, we said, is involved in fight-flight situations, which is the sympathetic, okay? That usually stimulates, and when you're doing physical activity, this is the one that's most active, okay? And I'll show you a very good example of how the brain works with, and how it involves all these systems, okay? And then, of course, the parasympathetic regulates conditions, usually at rest. That's why when you've eaten a big meal, you kind of sort of want to just fall asleep and just not do anything, okay? You don't like eat a big meal and then run a 100-meter dash. You don't do that, right? Because then you'll be trying to be doing all those, like, it puts you in a kind of sort of sleepy state. So you don't want to do that. And then, of course, we said the enteric only does, control of the digestive system activities. So you got all those divisions and subdivisions and how they are divided up and what they do. So you've seen this like many times so far. So which means I'm going to definitely want you to know this, and I'll ask about all these divisions and what they do and subdivisions. Okay? Know how it's all divided up and be able to describe the activities from that. So this is a very good example of the functioning of the nervous system, right? So what do we have here? That's the stimulus, that's the change in this person's environment, right? What is the change? Is the appearance of a bear or a lion, something that you might want to get away from. So what sensory input is he inputting from which organs or from which senses? Which sense is detecting this change or this stimuli? Huh? It's not a trick question. Or do you really smell the bear first before you see it? So you first see it, but yeah, you do smell it. Sometimes you smell it, and you can hear it. Yeah, it's true though. The reason I'm saying that is that I thought everybody's reaction would be sight first. You know what I'm saying? Rather than smell. Because we're not very strong at smelling, the humans are not. But other things like cattle, animals. But anyway, yeah, you do smell it. So that's one of the senses is smell, but obviously sight of seeing one. So, and all that information is conducted through sensory neurons, right? Like we just talked about. It's conducted through the sensory neurons, through the sensory division, and then it goes to the control center, to the brain, right? Where you're going to make sense of that information. Making sense of this information is saying to yourself, there's a bear over here and I need to get out of there. You need to get the hell out of here, right? So, yeah. And then you send instructions now to the via the motor neurons. Okay? So this is just like a homeostatic mechanism, right? There's nothing really new here. Okay? So you need to send to all your systems now. Okay? The skeletal muscle, of course, through the somatic being the most important. Why? Because you need to send a whole bunch of blood to your, quadriceps on the front, hamstrings on the back, so you can get out of there, right? So you're sending all that and oxygen and whatnot through here. So that's the effector. But what else do you need to do with the autonomic nervous system? You can do many things with here. First of all, you need to inhibit the digestive activity. If you are digesting your burger from McDonald's, you're going to have to stop that. Okay? Send all your blood to your skeletal muscles in the legs to get out of it. You stop that. What else do you do? In order to send all that blood, what do you need to do? You need to pump. Yeah, you need to send all that autonomic nervous system input through the cardiovascular center for your heart to beat faster. So your heart starts to beat a lot faster. So a lot of autonomic activity is going on there. Okay? And then you get out of it. And then that's your response. Getting out of there is your response. Okay? So this is a good example of how the nervous system functions. And, because it's emotional, you remember it. I know how it's something. Okay, so now we talk about the cells. We've talked about how it's all organized, how it's all divided up, and how the functions are, divided up. So now we talk about cells. So we said there's two main new cell types, right? The neurons, okay, which are excitable because and they can pass on electrical signals, action potentials, right? Action potentials. So most of the neurons have like dendrites, you know what I'm saying? On the cell body, they look like stars. So dendrites, and then, I drew this when we were talking about the neuromuscular junction as well. And then one very long process, which we said does what? Branches profusely, didn't we? Okay, and it's got many terminals. Okay, and then it's got a nucleus and a whole bunch of those organs, and I'll show you a picture. Okay, and then this part here, which kind of sort of gathers. So you got the dendrites here that gather information from the surroundings and send information towards the cell body. All right, and then that information is kind of gathered here by the trigger zone or the axon hillock, and then before it is propagated along the axon to go to wherever it's going. So those are neurons, okay? And neurons, because they don't have centrioles, they don't divide, okay? So actually, you're born with a certain number of cells in your brain, and that's it until you die. In fact, the number starts to go down, especially if you don't use your brain. That's why they say do crosswords puzzle or start when you're old to keep exercising your brain. Otherwise, you lose your neurons, and once you lose them, you cannot regenerate. Okay? So, because neurons are so important and they don't divide, and you need them, you have a lot of support cells called neuroglial cells. For short, you can just say glial cells. Okay? And I won't say no abbreviations in the example, because glial cells is a well-accepted abbreviation for that. So, neuroglial cells support and account for over half of the weight of the brain. It's only neuroglial cells, and the rest are neurons and a little bit of extracellular space. Actually, in the cerebrum, the cortex is the upper layer, which is only about 5 millimeters thick, which is about this, and it's pegged with 3/4 of all the neurons in the nervous system. Actually, that's the gray matter which surrounds your cerebral hemispheres. Okay, and then the rest of them will be these support cells. Okay, so neurons and neuroglial cells, and then a little bit of extracellular, space. But for the most part, they are really crammed up in there, especially the cortex, like I just said, in the cerebral cortex. All right. So know these two cell types, neurons and glial cells. Know what they do. Okay. So we'll talk a little bit more about neurons, because we said neurons are very special in that they do not, regenerate. Okay, so they receive stimuli and they transmit. So the dendrites, like I said, receive stimuli from the surrounding and then transmit it through the axon. Right? So we know about that. And then, so there are three components. Remember when I drew it, I drew sort of something circular, which is the cell body that contains all the organelles in it, including the nucleus, and then of course the dendrites. Those shorter processes that I said conduct information towards the cell body, and then one very long process called the axon, which is what generates, the electrical impulse that will go to the whatever the destination might be, whether it be controlling a muscle in your big toe or control another neuron, which are called interneurons in the brain and so forth. Okay, so pretty much the axon is an extension of the cytoplasmic, containing the cytoplasm, and it's just a membrane that transmits the impulse via action potential, right? And these axons, we'll talk about it a little later, can be myelinated, which means covered by a white sheath that will shorten the neuron, because that part that's myelinated does not conduct impulse. Okay, and I'll show you in a minute. And in fact, it speeds up transmission, because only the part that's not myelinated, called the nodes of Ranvier, are the ones that will transmit the impulse, so it kind of jumps. Okay, so these are the three parts of the neuron. You need to know them, and you need to know to be able to describe them and kind of sort of give a function for each. All right, give a function for each of these parts. All right, so I drew this before. This is what it is. And this is an example of a multipolar neuron. Why? Because it's got many dendrites, many processes in one. And then I'll show you later on pictures of the other types of neurons. Okay? Like I said, you got mitochondria in there, you got some endoplasmic reticulum, then you got a whole bunch of these mesel bodies with free ribosomes and endoplasmic reticulum for protein synthesis. There's so much of those endoplasmic reticulum that we call it mesel substance. See this mesel substance? And like I said, right here at the base of the axon is the axon hillock or trigger zone. You can call it whichever you prefer, you know what I'm saying? Why does it do that? It's because it's magnetic, you know. So if I can make it high enough. Axon hillock, okay, or you can call it the trigger zone. So all the dendrites transmit their, action potentials here before they are propagated along the axon. And I talked about myelination, right? So these are the myelin sheath. You see one myelin sheath here, and the space between the myelin sheath is what's called nodes of Ranvier. Okay. So when an impulse is being conducted on a myelinated axon, the impulse is going to jump only to the nodes of Ranvier, and that speeds up transmission to the tune of from 1 meter per second in an unmyelinated axon to 150 meters per second in a myelinated axon. So that's a smart. You know what I'm saying? Because if you step on a tack on your big toe, you might want that information to go quickly to your brain so you can withdraw before you damage a lot of tissue. Okay, so most of those axons, which can come from a millimeter long, a few millimeters long, to 1 meter long of axon. Okay. Another thing about the axon is that it has these branches at right angles called collateral branches, but these ones are very rare though. Usually it's just that one long axon. So very rare do you get these collateral branches. Okay? And they are at right angles to the axon. Okay, so it happens very rarely. Somebody in the morning asked, why do we have collateral branches? And I thought that was a good question. But usually, and I gave them an example of imagining a neuron controlling some of your thigh muscles. The thigh muscles, they are very huge, right? Many cells. Even though it branches profusely, you might want sometimes a collateral branch that will also then branch, so you can control more. So the complexity of the organ being controlled by the neuron sometimes will determine whether there's a collateral branch or not. Okay? So in the exam, if I ask you why do you have collateral branches in some types, or give me a reason for them, you will be able to tell me, right? Okay, good. Because I saw some of you yawning and sort of, I gauge those things, and those are the things I ask you in the exam. Okay? Okay, so now we talk about the soma, because the soma contains, or the cell body of the neuron, contains all these organelles we've been talking about, right? So it contains the nucleus, of course, which is very important because it will conduct and direct all the protein synthesis that must go on to repair, especially when a dendrite falls off, for instance. Because the cell is not going to regenerate, it will need to make proteins and so forth to patch up or to fix that breakage, since these are not going to regenerate. So we have a lot of nasal substance, and nasal substance is a lot of rough endoplasmic reticulum and a bunch of free ribosomes. All right. What do free ribosomes make? Remember from the organelles that we did, and it was in the exam. What do free ribosomes make? What's that? Yeah, the proteins that are used inside of the cell. So you can imagine how much proteins are going to be used by that neuron, which does not regenerate, so it will need to fix itself by creating a whole bunch of these proteins. So that's why you have a whole bunch of these free ribosomes and a rough endoplasmic reticulum, which again is the site of what? The rough endoplasmic reticulum? Protein synthesis. Okay, so you need a whole bunch of that. And there's so much of it, we have a special name for it called nasal substance. You know, sometimes in the exam, I'll give you a multiple choice question and say nasal substance in neurons is made up of, then I'll say go to and then you know what to say, right? So, also there's a Golgi apparatus in there, a lot of it, you know, because the Golgi apparatus does what? Yeah, it packages and gives tertiary structure to these proteins and so forth, and how they are folded up. If it's going to be a glycoprotein, for instance, it's going to be attached to the carbohydrates in the Golgi apparatus. Okay, so you have that to deal with that, and of course you know what this does, which is all the energy, and a whole bunch of other organelles are present. But these are the important ones for the protein synthesis and all that, to create all these repair parts for these neurons, since they're not going to regenerate. Why don't they regenerate? Because they don't have these. Centrioles, what is the function of centrioles? Since we've done mitosis breaking down of down 110. What is the centriole do? What is the centriole? What is function is it involved in the cell?
The other inputs and their extensions of the
Yeah. That's right. That's good. You're the one who was able to answer that question. You see, when the chromosomes are put in the center, at the equator, before the cell division, and do that, and this is pulled up by those fibrils like that, it is the centrioles that put them, line them up like that, and move those chromosomes to where they need to be. So if a cell does not have centrioles, you know that it's not going to be able to divide, right? Because it has to do that in order for it to divide. So the reason I'm talking about that is that if you know, if somebody tells you something doesn't have a cell doesn't have a central, then in your head, without even thinking, you're able to say it doesn't divide. Which is what I want you to do with all this information, because with this physiology stuff and in this course, it kind of builds up. We talked about cells at the beginning and talked about all the organelles. And then when we were in muscles, we pretty much stripped up all the organelles and we were left with myofibrils because it contracts. Okay, and this is specialized for what it's specialized, and you have all these other organelles in there. So it's pretty much the same stuff, but we're just compartmentalizing it into the different functions of the body and how it those things work, those parts of the body work. Okay, so pretty much when you have clusters of cells, neurons, in the peripheral nervous systems, those are called ganglia. Okay, and if you have clusters of neurons, cell bodies, in the central nervous system, those are called nuclei. Okay, not the nucleus of the cell, but they just called nuclei. Okay, nuclei, and it's spelled exactly like the nucleus of the cell, but it means just groups of neuron cell bodies in the CNS. Okay? So that's that. So there's more about neurons that I'm going to talk about. Like I said, you see the axon hillock, or the trigger zone, what they were calling the trigger zone. Okay, so you can call it whichever you like. Now, I like these diagrams because they highlight a particular thing, so you can see it in relation to the rest of the stuff. So you can look at that, and then the dendrites, of course, the nucleolus and the nucleus, and the axon. Okay. All right. So there we go. So neuron structure, we've kind of sort of talked a little bit about it. Okay, and this is the hillock or the trigger zone. Okay, and this is the part that generates or gathers all the input from the dendrites before you generate an action potential in the axon. Okay, so that's what it does. So sometimes I would say, tell you to describe the functions and characteristics of neurons. Okay, and then you talk about all this stuff. Sometimes I specifically talk about the characteristics of it, and you talk about the trigger zone, the dendrites, the cell body, and the axons, and sometimes those collateral branches. So the axon is pretty much a slender process that is the longest, okay, and it can be a millimeter long, a few millimeters long, a centimeter, up to 1 meter, right? Imagine a neuron that's starting from your cerebral cortex and controlling your big toe. That's going to be like 1 meter long, especially if you're a shark. It's a couple of meters long, you know what I'm saying? Shakir is tall, so he's going to have one long axon to get to his big toe from the head. So it can go up to as far as 1 meter, okay? And like I say, all those neurons, they branch profusely so that they can supply more structures. So there's usually that one long process that we talked about called the axon, and it's usually unbranched. Very rarely do you get those collateral branches I was talking about. And remember the situation I kind of described, that will kind of sort of necessitate having a collateral branch. And it will kind of sort of depend pretty much on the complexity of that part that's being controlled by that neuron. It's very complex. You may have a bunch of branches, terminal branches, but if you have a collateral branch, then you can kind of control more things with the same neuron for the price of one. You see what I'm saying? Isn't that good? So, like I said, you get up to 10,000 branches, but there are those few instances when you might need a collateral branch, the one that is at right angle to the axon, and then it will also branch and then be controlling more things, okay? And we already know about the synapse, right? What is the synapse? What did we say synapse is? Yeah, it's a functional connection. I like that because in the morning, maybe they did. In the morning it's too early sometimes to think. You know what I'm saying? So functional connection. Because why is it a functional connection? Anybody? Because there's no physical connection between the two. In fact, there's a gap which we call the synaptic cleft. So between we talked about the instance of a neuron and a muscle cell. But in this instance, it is mostly neuron to neuron, because the neurons will communicate with each other to pass on information. And there's some neurons that are specialized to process a certain action during integration, like those neurons. So once the information hits the brain, it's going to be triaged to the particular group of neurons that best deals with that sensory information, right? So all those will be synapsing with each other. They're not touching, they're synapsing. And okay, we have other neurotransmitters in the brain like dopamine, for instance. Especially when you do drug abuse, dopaminergic neurons that are affected when you're high, right? Not that you guys have been high. You know what I'm talking about? Because dopamine gives that pleasurable feeling, it gives you that high. The dopamine neurotransmitter, okay? So anyway, so like I said, if you have, bundles of axons, they are grouped like that. In fact, when we do the muscular system in the cat, I'll be showing you nerves, okay, which are bundles of axons, and you'll be able to see them. They're pretty thick, actually, okay? And they're called nerves when it's in the periphery, and they're called tracts when it's in the CNS. In fact, there's connecting your left cerebral to your right cerebral hemisphere. There's like 300 million, axons crisscrossing from this side to that side of the brain. So that the population of the whole of the United States, and you have axons, crisscrossing across, and those will be tracts. Okay, so those are tracts, and they are white in appearance because they are myelinated. Okay, and I'll show you a picture at the end of the brain and these tracts and so forth. So nerves when they are outside of this, CNS, and tracts when they're in the CNS. Okay, and then glia, ganglion when they're in the periphery. Those are groups of cell bodies, and then nuclei when they are in the central nervous system. So you got to keep that in check, okay? And so forth, okay? Very good. All right. Okay. So now, like I was talking about the types. So far we've been talking about multipolar, right? The one I drew on the board, the one I showed you, the diagram of all those are multipolar. Why? Because it's got a lot of dendrites, those shorter processes, and one axon. Okay, so those are multipolar. And most of the interneurons, like I was talking about, the ones that connect one part of the brain to another part of the brain, those are interneurons. All the motor neurons are multipolar because those ones control, the big leg muscles and so forth and so forth. So they got all sorts of branches, and then they got all sorts of dendrites. Okay, so they can connect and gather information from many parts of the brain. So multipolar are the bulk of the neurons in the body. The nervous system is mostly multipolar, right? Makes sense? Okay? And there are several dendrites, a lot of dendrites, one axon. And then bipolar, just like the name says, two processes. All right? And I've been drawing multipolar up to now. So the bipolar will have a cell body, boom, boom. Okay? So one of the processes will act like a dendrite. One of the processes will act like an axon. Okay, so that's bipolar. And this is mostly reserved for special senses, like smell, olfaction. Those are bipolar neurons. Sight, it's got bipolar neurons for the special senses. So most of those are sensory. And then, of course, unipolar. Unipolar is just one process, okay? This is a little bit like that. So you got the cell body over here, and then you got a connector here. And then pretty much the part that kind of acts like a dendrite, that kind of has sensory information, is called, the peripheral branch. Peripheral branch. So this is kind of like the sensory part, okay? Okay, and then this part here that connects to the central nervous system is called the central branch. Okay? Okay. So this is kind of like a dendrite that gathers sensory information and passes it along. So it's one process, yes, but it's got a part that sort of gathers information called the peripheral process, and then the peripheral branch, and then the central branch is the one that will conduct the information to the central nervous system. Okay, so that's unipolar. Again, mostly sensory for the special senses: sight, hearing, balance, and so forth, and things like that, and smell. Okay, so we know the three types of neurons. We know the two major cell types: the neurons and the glial cells. Okay, we know a whole bunch of properties of the neurons. Okay, and all the kind of organelles that they have. So multipolar, mostly interneurons and motor neurons, and then bipolar, special senses like smell, sight, and hearing, and things like that. And then, of course, unipolar, also for special senses. So in the exam, I might ask you, okay, can you give me an example of all these types of neurons and what they do, or where they're most likely to be found? Okay, and you'd say multipolar, these are mostly interneurons, okay, and mostly motor neurons. Okay, and then you talk about bipolar, you say these are mostly in special senses like sight, smell, hearing, and so forth. And then the same with bipolar, unipolar. Okay, so those are the things I'll ask you in the exam. Okay, so that's a 2-point question. You get the third one for free, for bonus. If that makes sense. Okay, so glial cells. Now we said there's 2 glial cells in the peripheral nervous system, and those are Schwann cells and satellite cells. And then we got 4 in that central nervous system, right? So what do? We talked about it. More than half of the weight of the brain is pretty much glial cells or neuroglial cells. All right, they provide support. They scaffold neurons, and they kind of sort of, surround blood vessels and surround neurons and kind of bring the neurons close to the blood supply, nutrient supply. So they kind of bring them to the nutrient supply and kind of shield them like that. And they also segregate and insulate neurons. Remember, neurons, what are they doing? They are passing on electrical signals. Okay, just like the wires in the house. Have you seen the wires in the house, electrical wires? Do you ever see them bare? They're always covered by a rubber. So, you know what I'm saying? Same thing with the neurons. They serve that purpose to insulate and electrically insulate these neurons so that they don't cross-contaminate and pretty much not cross-excite other, neurons and stuff. So, when especially you are creating the nervous system, especially in the fetus or whatever, and young neurons are growing, and so they are guided by these glial cells so that they grow into the right spot. For instance, if it's supposed to grow to your big toe, it's gotta go there. Okay, so this guides them, those young neurons, so they grow into the correct spot and they connect the correct things. So they have the proper connections. Okay, because sometimes I do ask about functions of neuroglial, the general functions of neuroglial cells. Then I also specifically ask you about certain neuroglial cells, and I want you to talk about it, and we'll be talking about it in a second. And they also promote health and growth of these neurons. Okay? So many functions for these glial cells or these supporting cells. Okay, glial cells do that. And we said there's 4. Astrocytes, you can see by the name what kind of shape is it? Star, because Astra is star. You know what I'm saying? So Astra is a star. So these are sort of like multipolar. They look like multipolar. Okay, and these ones do a lot of things. They're involved in the blood-brain barrier. Okay, the blood-brain barrier, which only lets certain substances into the brain because you don't want to contaminate your control center. And then you got a lot of things like repair, things like that, and we'll talk about that. Okay. And then you got microglial cells. What kind of cells would those be? Just by the name of it, microglia. They're small. They're minute. That's why microglia. They're small. They've got few processes, and these are the macrophages of the brain. What do macrophages do? Eat off bacteria. So that's very good. Okay. So these are the sort of the immune cells of the brain. Okay. So ependymal cells. This one doesn't kind of say what it is, but these are pretty much sort of columnar cells with a cilia on them, and they are involved in creating CSF, okay, cerebrospinal fluid. So these will be lining the ventricles, which are chambers in the brain that contain cerebrospinal fluid, right? And also cerebrospinal fluid, where do you find it? There's 3 layers of membranes that cover the brain, right? The one which is the toughest on the outside is the dura. Then the inside one is the arachnoid, which looks like a spider web. That's why it's called arachnoid. And then the pia mater, the innermost. So the subarachnoid space is also filled with cerebrospinal fluid. Like your brain is cushioned by that. In the morning, I was talking about how when you're drinking, you start to go to the bathroom a lot, you get dehydrated. So drinking alcohol does that. It causes dehydration. So you end up losing some of your cerebrospinal fluid. And that subarachnoid space is no longer having enough fluid to cushion your brain against your cranium. God forbid you're driving and you have an accident. What happens is that the car stops. If you wreck on something, the car stops, but your brain and your thingy continue to move, right, in your car. So and then because of less cerebrospinal fluid, your brain pretty much crashes against your skull, and you get really serious damage on it. So don't look at that.
What was the astrocytes are the
or the blood-brain barrier. And I'm going to show you again, I'm just sort of preempting the information, but I'm going to show you a list with all the information of astrocytes. So it's blood-brain barrier. Okay, the blood-brain barrier. And the blood-brain barrier is very important because it only lets things like glucose in there, because the blood will only use glucose for energy, nothing else. Okay, so that's why when you get low blood glucose, what happens to you? You become faint, right? Especially people who have diabetes and they're not regulating their blood glucose and it fluctuates and they get too much insulin and they don't eat, they get into a coma because there's not enough glucose to go in there. Okay? All right, so that's what it does. And then anyway, the ependymal cells, they make CSF, and then of course oligodendrocytes. Oligo means few. They got few dendrocytes, and these make the myelin sheath. Remember the myelin sheath that I was talking about that surrounds those axons? It's made by the oligodendrocytes. Okay. So those are the 4 in the central nervous system, and then there's only 2 in the peripheral nervous system. So the satellite cells usually just sort of surround the cell bodies and kind of support. And then the Schwann cells, what do? They create the myelin sheath, but in the periphery. Okay, so they are just the counterparts of oligodendrocytes, the Schwann cells. Okay, they are in the periphery and they create it. So we're going to look at each and every one of these cells individually and talk about it. So the astrocytes, of course, are the most abundant and they have the most functions because they are the most abundant. So they are versatile, they are highly branched. Okay, that's why they are astrocytes, star-shaped. Oh my God, this is a poor star. Okay, so what do? They cling to neurons and they sort of, cling to blood vessels as well and kind of bring them to close proximity to each other so that the neurons are very close to the blood supply and the nutrients and the oxygen, okay? So that's what they do. And so the functions are mainly, like I said, supporting, bracing neurons in place, okay? And I'll show you a nice diagram that shows the function of astrocytes. And then, of course, cranial blood vessels close to the neurons so that they're close to the nutrient supply. Okay, so they anchor neurons right to the blood vessels right there. Boom. Okay, and then, of course, like I was saying there earlier, influencing the blood-brain barrier, the 3 B's. Blood-brain barrier. It's kind of like a tongue twister. So the blood-brain barrier is very important. And in the morning I was talking about how in the pharmaceutical industry they try to find drugs, especially for psychological conditions, that go to the brain. And that's very hard because of the blood-brain barrier, which is created by the astrocytes. It's like a guard. It doesn't let anything through there. You can imagine your control center that controls all the activities in your body. If it gets compromised, then so will you. You know what I'm saying? All your heart rate, everything goes haywire. It's no longer controlled properly. Okay, so the blood is very selective what it lets up there. So that's one of the big problems in the pharmaceutical industry, trying to find drugs that do that. Because I used to work in there, and I used to research on drugs for schizophrenia to go to the brain. So they have a process where they screen a whole bunch of compounds called high-throughput screening, just in order to beat this. You know what I'm saying? Where they screen a lot of compounds to try and see which one goes up there. Because not only do you want it to have efficacy to do the job, not just enter the brain and sit there. You see what I'm saying? So you want it to get into the brain and do stuff. So the blood-brain barrier really lets almost nothing except glucose and a few other, molecules in there. It's very important too, because you don't want to compromise. And then it processes substances. Okay. In the morning I was saying, when you clean up, when you cook a meal in the kitchen, you are sloppy sometimes and you sprinkle stuff, and then you tell your kids to come and clean up the kitchen. So those will be your astrocytes, your kids, because they do the same thing. Like neurons are busy conducting impulses in and out, and that's ions moving into and out of the membranes. And so sometimes you get all this potassium leaking and potassium all over the place. Astrocytes come in and mop up all those excess ions so that it remains all good, right? So that's what happens. So when the neurons do their business of conducting impulses, the astrocytes come in and clean up, make sure that the environment is all good, and also recycle neurotransmitters. Remember we talked about how the acetylcholine is recycled at the neuromuscular junction, and we talked about acetylcholinesterase. So this will be that. They don't take that function. The astrocytes will do that. Because you can't be releasing all these neurotransmitters and you're not replenishing them. You soon run out. Okay? So you need to recycle them. So if I ever ask about functions of astrocytes, which I normally do in the exam, we talk about all this stuff. And they're easy to kind of think about when you think about what the neurons are doing and all these things. Think about all that and the support that the neurons need, especially that they can't divide. Right? Sometimes they will isolate damaged parts, and you really need to have a good repair process, especially if you're not going to be building new houses. The house that you have, you're going to be plastering all over it, rebuilding, and that's the same deal with these astrocytes. They'll do that, isolate damaged parts, and repair. So they're involved in repairing the neurons, especially that they don't divide. So, gotta know all these functions of astrocytes in there. You see a good diagram showing you how astrocytes work. You see how you got the cell body here, and then this is a neuron, and then you got an astrocyte, and it's holding, cradling, the anchoring pretty much the neuron to the blood supply, so that it's close to the capillaries, providing oxygen and so forth, and nutrients. Okay, so ependymal cells. Ependymal cells, we talked about how they're involved in creating cerebrospinal fluid, and we said they're mostly columnar cells that have cilia on them. What does cilia do? Remember when we talked about the organelles, it moves what? Yeah, because anything on the top of the cell surface, like this, cilia will be involved in moving and circulating cerebrospinal fluid, okay, in those ventricles and so forth. Because it doesn't just sit in there, it's circulated. Okay, so, and we all know about squamous cells, right? Some of these cells are squamous, and squamous is what kind of cell shape? Flat. So you all know about that. And then columnar with cilia, just like this, to move and circulate cerebrospinal fluid. So they are special ependymal cells that kind of line up blood vessels that are called choroid plexus. And those are the ones that make, and those are usually suspended inside of the ventricles, and they create cerebrospinal fluid. When we do brain dissection, you'll be able to see these, specialized blood vessels lined with ependymal cells that create the cerebrospinal fluid. Okay? Because the function of it is to produce and circulate cerebrospinal fluid. That's the ependymal cells. Okay? And in the exam, usually bring about questions to asking some of these glial cells, where they are located, what they do. Okay? I usually do that. And then, so my problem already said they are small cells, few dendrites, and they are the macrophages of the brain. And they engulf bacteria, they are phagocytes. Engulf foreign microorganisms and destroy them. Okay? So those are microglia, and the name will give you characteristics for the most part, except the ependymal cells. The ependymal cells are the ones that don't give away the structure. But you sometimes you win some, you lose some. Okay? But because I would expect you to know the characteristics of these cells, and I would expect you to tell me about the functions of these cells. Okay, these glial cells, right? And so oligodendrocytes, we talked about it. We said they create the myelin sheath. What is the myelin sheath? Pretty much, it's an extension of that cell and a bit of cytoplasm that will surround and circulate around that axon, and it will insulate that part, which you see how this kind of extends and it circulates around and it wraps around that axon. Okay, and then of course there are parts that are not circulated by that, which is the nodes of Ranvier. Okay, and those are the parts that contact the impulse, right? Nodes of Ranvier pretty much shortens, okay, the impulse. I mean the axon, so that the impulse jumps from one, node of Ranvier to the next, and that's called saltatory conduction of the impulse, when it jumps from one node of Ranvier to the next node of Ranvier. Okay? And oligodendrocytes produce the myelin sheath in the central nervous system. Now we go to the peripheral nervous system, and we have just the two cells, satellite cells and glial, and Schwann cells. So Schwann cells, we said they are the counterparts of oligodendrocytes in the CNS, and they create the myelin sheath in much the same way, just wrap around these axons to create the myelin sheath in the peripheral nervous system. Okay, and then of course satellite cells, they kind of do the same job like the astrocytes in terms of kind of sort of supporting and nourishing. Usually they surround the ganglia. We said ganglia are accumulation of cell bodies in the periphery. So that's where you find the satellite cells. You see all the satellite cells, cells surrounding the cell body. Okay, and then it's called ganglia in the. So of course, you know these two cell types in the peripheral nervous system is glial cells and what they do. Okay, just like you need to know the 4 types. So myelination, I was talking about it, and I said it pretty much insulates. Okay, because it's whitish in color. Okay, it's got fatty acids in there. It's got protein and lipids. Parts in there, and it insulates because it doesn't conduct. Okay, pretty much like I was saying, it's an extension of that cell, and together with a little bit of cytoplasm surrounds the axon. So that's pretty much what it is. And it will be a swan cell doing that in the periphery, and the oligodendrocyte doing that in the central nervous system, and that's it. And it forms a myelin sheath by winding itself repeatedly around the axon. Okay? And we said this is what gives the white appearance. That's why we end up having white matter, which will be myelinated axons, because only axons can be myelinated, nothing else. But the gray matter is going to be unmyelinated structures, because it can be many. Dendrites are not myelinated, cell bodies are not myelinated, and some axons are not myelinated. So that's what you have to say if I want to talk about that in the exam. So functions: protects the axon, and it of course insulates. All right, it electrically insulates not only that part of the axon, but from other axons as well. That's what I'm saying, so that they don't cross-excite each other. All right, and then, like I said, it will increase speed of transmission to the tune of 150 meters per second. Okay, from 1 meter per second in an unmyelinated axon to 150. That's just amazing speed. Okay, if I step on a tap right now on the barefoot, I'll be able to feel it almost instantaneously. That's how fast it is. Okay, and we said the spaces that are, or the gaps where there's no myelin sheath, are called nodes of Ranvier. And those are the only parts of the axon on a myelinated axon that will conduct impulse to give that saltatory conduction that I just talked about. Okay, which is saltatory means to jump. You know, the impulse will jump from one node of Ranvier to the next, and therefore pretty much shortens the axon. So know about myelination and unmyelination. Okay, so unmyelinated axons do not have myelin sheath on them, but there are still axons that do what? Conduct electrical impulses. Okay, so they're not going to be bare wires, just like you don't find any bare wires, electrical wires in your house or in the car. They have to be insulated. So the same cells, the oligodendrocytes in the CNS and the Schwann cells, you see how they kind of sort of wrap around, and sort of, you see how 1, 2, 3, 4, 5. These axons are all separated from each other by the cell. It doesn't wrap around like a myelin sheath does, like as if it was a myelin sheath. It will only sort of insulate and against each other, so that they don't like excite this one, number 2, number 3, and so on. Okay, so that's the function of the Schwann cells and the oligodendrocytes. Okay, apart from creating the myelin sheath, they also insulate these unmyelinated axons, because they're still conducting electrical impulse, and they need that. Okay, so they do that. So those invaginations of the, the myelin cell, the Schwann cell here is shown, and also the oligodendrocyte. Oligodendrocyte in the CNS. Okay, so you can see the myelinated one on the top, And you see the difference with the one that's not myelinated, but still insulated by those glial cells, Schwann cells and oligodendrocytes. Okay? So, because they're still connecting. Is it not going? Okay, so, more organization of nervous tissue. What do we got here? Like I said, myelination results in formation of white matter. Okay, all those tracks we talked about that connect different parts of the brain, okay, are all like myelinated axons, and that make up the white matter of the brain. Okay, and white matter of the spinal cord as well. Okay, so propagates action potentials, high speed, consists of myelinated axons, forms nerve tracts like I talked about in the central nervous system, and nerves in the periphery. Okay, so those are groups of neurons bundled together. In the periphery, they're called nerves, like spinal nerves, cranial nerves, and in the central nervous system, they're called tracts. Okay, so any other structure that is not myelinated forms the gray matter. So you've got cell bodies that don't get myelinated, you got dendrites that don't get myelinated, and then you get some axons that don't get myelinated. All that together form the gray matter. Like I was talking about, the cortex, the cerebral cortex, which I said is packed with 3/4 of the neurons, 75% of all the neurons in the nervous system are located in that layer, the cortex, which is like 5 millimeters thick, and all those neurons are packed in there. Okay, that's what forms the gray matter on outside the cerebral cortex, there are also some nuclei on the inside that are, gray matter as well. And then, of course, ganglia on the outside, on the periphery. Okay? So, that's that. And then, boom. So, axons, they will synapse with other axons and cell bodies and so forth. And that's where the integration takes place. When one neuron takes information to another neuron, those are interneurons, and they connect together in synapses and process sensory information that way. Like I said at the end, I was going to show you this, which is the spinal cord, and you see how it starts off with the dorsal root. The dorsal root is carrying sensory information towards the CNS, and this ventral root carries the motor information. And there's a synapse here of the dorsal root, so it forms the dorsal root ganglia. Anyway, the inside will be the gray matter on the spinal cord, right? And then the outside is the white matter, so you have tracts there of myelinated axons going up and down also, okay, on the spinal cord. So that's white matter. And if you do the brain, the same thing. So this will be the gray matter over here, which will be the cerebral cortex. Like I said, it's a very thin layer, not more than 5 millimeters thick, and that's where all the neurons, about 3/4 of the neurons, are packed in that gray matter, neuron cell bodies and so on. And then, of course, the tracts. You see these tracts, the white matter. So this is the corpus callosum that I was talking about earlier on, where you have 300 axons crisscrossing from the left to the right side. So that's how your two cerebral hemispheres are connected by those tracts. Okay? Boom, boom, boom. That's it, folks. Thank you so much. Remember, we are in 417, okay? And we shall be meeting there, okay? For the electrophysiology. Thanks. We shall see you soon.